Hello friends, welcome to this lecture Growth Phases and Kinetics in Plants. During this lecture, our main objectives will be to discuss the process of plant growth, explicate the mechanism of growth kinetics in plants, study the various phases of growth and development, and lastly we will be studying the various processes related with plant growth. Let's start with the introduction. The term growth can be defined as a permanent and irreversible change in the size of a cell, organ or whole organism, usually accompanied by an increase in dry weight. Growth in plants mainly occurs by the activity of meristematic cells. The new cells are continuously produced by cell division in a meristem. Apical meristem lies at the apex of every shoot and root, known as root apex and shoot apex respectively. Some plants have intercalary meristems. Grasses, for instance, have such meristems at the bases of internodes. The vascular cambium is another kind of meristem which by its activity causes increase in the girth. The daughter cells derived from cell division into meristems enlarge and bring about visible changes in the size of the plant. The expanding cell differentiate to form the different tissues and become part of the mature organ. New cells form continuously at the apical meristem. Cells enlarge slowly in the apical meristem and more rapidly in the subapical regions. The resulting increase in cell size can range from several fold to hundredfold, depending on the species and the environmental conditions. Tissue growth is neither uniform nor random. The derivatives of the apical meristems expand in predictable and site-specific ways, and the expansion pattern in these subapical regions largely determine the size and shape of the primary plant body. The total growth of the plant can be thought of as the sum of the local patterns of cell expansion, while the morphogenesis, that is the acquisition of shape or form, is the sum of the patterns of both cell division and cell expansion. Plants are unique multicellular organisms that retain the capacity for unlimited growth, that is, an overall increase in size throughout their lives. They are able to do so because they have meristems at specific locations in the body. Meristems are composed of stem cells which perpetuate themselves by cell divisions and also give rise to derivative cells which differentiate along new lines. As a result of meristematic activity, fresh tissues and organs are formed and the plant continues to grow in height and in many cases girth throughout its life. This form of growth in plants is called open form of growth. The advantage of this type of growth for organisms that are rooted but subject to predation are immediately obvious. Plant meristems can be classified on the basis of location. Based on this, three main different types of meristems have been reported. Number one is the apical meristem. It's located at or near the tip of shoot or root and are called shoot and root apical meristems. They are responsible for primary growth, including elongation growth of these organs. Initiation of growth is by one or more cells situated at the tip of the organ which maintain their individuality and position are known as apical initials or apical cells. Number second, we have the lateral meristems. They are located on the sides of roots or stems and add to the girth or secondary growth of these organs. Vascular cambium and cork cambium are the examples of lateral meristems. Lateral meristems are composed of initials that divide periclinally. And lastly, we have intercalary meristem. Intercalary meristem typically occurs intercalated between mature regions as at the bases of grass leaves. However, in a broader sense, they can be considered to occur at the bases of leaf primordia and above the nodes in stems of nearly all plants and add to the elongation growth of these organs. In addition to these three types, meristems are also classified as indeterminate and determinate. Shoots and root apical meristems and vascular cambium remain active throughout the plant life. Hence, they are referred to as indeterminate meristems. In contrast, meristems involved in the leaf and flower development are active only for a short time and are used up in the formation of these organs. They are called determinate meristems. After having discussed about the growth and its different means, let's discuss about the phases of plant growth. There are 3 types of growth, with cell division, enlargement, and lastly the maturation. Let's take the cell division first. A cell is metabolically highly active at the time of cell division. Its cellular mass increases and a replication of genetic material takes place. Growth as a result of division is based on mitotic cell division. In the stage of mitosis, each chromosome splits lengthwise into 2 homolog chromatids, which then pass equally into daughter cells. Certain cell organelles with chloroplasts and mitochondria apparently duplicate themselves. As a result of division, each of the 2 daughter cells is only half the size of the plant cell. These cells then proceed to enlarge and synthesize their own cytoplasm and cell wall material. Now, after cell division, cell enlargement takes place, and it is before the cell maturation. The cells increase in size mainly due to vacuolation, that's by the absorption of water. A big central vacuole appears which pushes cytoplasm to be limited to a thin boundary layer against the cell wall. The new cell wall material is synthesized to cope with the enlargement. It appears that cell enlargement due to vacuolation is governed by osmosis. In addition, synthesis of new cytoplasm and cell wall material also accompanies the cell enlargement. Then after cell enlargement, cell differentiation is followed and leads to the development of specialized mature tissues. For example, the meristematic cells of leaf epidermis differentiate to form guard cells, trichomes, glands or epidermal cells. The differentiation and maturation of various kinds of cells depend on the number of internal, external, nutritional, genetic and hormonal factors. Thus, it is the final differentiation process in biological systems such as the final ripening of a seed or the attainment of full functional capacity by a cell, a tissue or an organ. After finishing with growth and the different phases of growth, let's move to the growth kinetics. Growth kinetics is actually the study of the growth and development of one part of an organism in relation to another. Many growth phenomena in nature exhibit a logarithmic or exponential increase. The size, mass or number increases by a constant similar to simple compound interest. Now, first let's take the absolute growth rate. If you plot growth, that size, mass or number versus time, a constant increase in the growth curve is obtained. If you calculate the slope between any 2 times, you get the absolute growth rate, which is the change in actual growth over time. You get a different slope, hence different AGRs for each pair of times chosen to calculate the slope. Next, we have relative growth rate. If you plot the logarithm of growth, that size, mass or number versus time, a linear line is obtained. If you calculate the slope of the line, you get the relative growth rate, which is the change in growth, relative growth over time. Since the line is linear, you get the same RGR regardless of which time interval chosen to calculate the slope. And next we have the sigmoidal growth curve. Exponential growth can never be sustained indefinitely. Eventually substrates are depleted, the population exceeds the area available, tissues or individuals begin to die. and so on and so forth, which decreases the growth rate. Growth may still increase, but at a reduced rate. It may reach a steady state or growth may begin to decrease. If you plot long-term growth versus time, you get the classical sigmoidal growth curve. If you plot the logarithm of the sigmoidal growth curve, you get a linear line during the exponential phase, after which the curve decreases over time. Thus, three main phases of growth are seen in the sigmoidal growth of plants, with lag phase, log phase, and stationary phase. Let's first take the lag phase. This is the longest of all the growth phases. During this phase, there is little or no cell growth. In a state of growth, the cells are busy replicating various proteins and DNA in preparation for the next phase. The cells are not shut down during the lag phase. They are very active metabolically, but are not getting any bigger. The lag phase is followed by the log phase, is also called as logarithmic phase. In this phase, the cells become extremely active and begin the process of dividing. Every chemical in the cell is being replicated in anticipation of the cell division. The log phase gets its name from logarithmic phase, which roughly means that there is massive growth. It is a period characterized by cell doubling. The number of new cells appearing per unit time is proportional to the present population. If growth is not limited, doubling will continue at a constant rate so that both the number of cells and the rate of the population increase doubles with each consecutive time period. For this type of exponential growth, plotting the natural logarithm of cell number against time produces a straight line. The slope of this line is the specific growth rate of the organism, which is a measure of the number of divisions per cell per unit time. After log phase, last phase we have to discuss is the stationary phase. The stationary phase is due to the growth limiting factor. This is mostly depletion of a nutrient and or the form of inhibitory products such as organic acids. A widespread explanation is that the stationary phase results from a situation in which growth rate and death rate have the same value. After growth phases, we move to the growth, differentiation and morphogenesis. Growth, as already said, is an irreversible increase in mass. Cell division by itself is not sufficient to result in growth. It is by differentiation that cells in an organ or tissue become different from each other or specialized for different functions. For example, the epidermis, mesophyll, xylem or phloem cells of a leaf. From a functional viewpoint, differentiation is equivalent to specialization. Morphogenesis is the acquisition of form. How a plant or organism acquires its distinctive shape or form. Because plant cells generally are fixed in relation to each other and they are cemented by the cell wall, morphogenesis in plants is essentially a function of planes of cell division and direction of cell growth. The control of these two processes therefore is central to the study of plant morphogenesis. Classically, plant growth has been analyzed in terms of cell number or overall size, that's mass. Growth in plant cells is mainly through the cell expansion driven by turgor pressure. During this process, cell increases in volume many folds and becomes highly vacuolated. Growth in plants can be measured in terms of number 1, increase in length or girth, number 2, increase in pressure or dry weight, number 3, increase in area or volume, and number 4 is the increase in number of cells produced. However, size is the only reliable criterion that may be used to measure growth. For example, growth can be measured in terms of change in fresh mass, that is the weight of the living tissue, over a particular period of time. However, the fresh weight of the plants growing in soil fluctuates in response to changes in the water stats. So this may be a poor indicator of actual growth. In these cases, a measurement of dry weight are often more appropriate. Cell number is a common and convenient parameter to measure the growth of unicellular organisms. But in multicellular organisms, cells can divide in absence of an increase in volume. For example, during the early stages of embryogenesis, the zygote subdivides into progressively smaller cells with no net increase in the size of the embryo. Only after it reaches the 8-cell stage does the increase in cell number begin to mirror the increase in volume. Since the zygote is an especially larger cell, this lack of correspondence between an increase in cell number and growth may be unusual, but it points out that the potential problem in equating an increase in cell number with growth. So dear friends, today we dealt with the process of growth in plants, different phases of growth, and lastly the growth kinetics. Thank you for being with me. Have a nice time.